All right, can somebody confirm you can hear me? All right, can somebody confirm you can hear me? Let me check my volume. Volume looks good. All right, let's begin. I've got too many Zoom windows open, so what's happening is the Zoom is making the lecture file, and so my computer is going to be working slow for the time being. I am recording. Let's share the screen. All right, you should be seeing my screen now. Today is November 14th, and you should be working on your unknown project and lab 9 today. You'll note there is no class for Wednesday. Any question about any of that? All right, let's go to lab 9. Trying to think. I think I'm going to close this down because I've got my computer is overworked. Actually, let me close that down too. All right, lab 9, the identification of gram-negative bacilli. This is the last lab for this course. You need to complete lab 9 identification of gram-negative bacilli, complete the case studies, and complete the lab 9 worksheet. There is some reading as normal that you should do before each lab. Boy, I don't have, there it is. My mouse here. Blow that up just a little. There are some video clips in today's lab. Upon successful completion of this lab, students should be able to, 1, interpret the results for the following biochemical tests. First, the Heimlich reactions. Heimlich is an acronym for 4 different tests. First, I for indole, M for methyl red test, V for Voges Proskure test, or the VP test, and C for the citrate test. And then the I is just put in there to make it a word. Heimlich, sort of a word, something you can pronounce. We're also going to talk about the carbohydrate fermentation test, which I think we've talked about in a previous lab, and the motility test and the hydrogen sulfide production test. So know all of these tests and how to interpret them. And then we'll also look at and explain how to interpret the triple sugar iron test, or the TSI test, the SEM test, that's the sulfur indole motility test, and then look at the growth on McConkey's agar and Hectone enteric agar. And McConkey's is abbreviated MAC test, and Hectone enteric HE agar plates. Any question about any of that? So use biochemical analysis to solve case studies. You'll have case studies in today's lab involving gram-negative bacterial infections. So in the gram-negative bacteria, many of the infective bacteria that cause gram-negative infections belong to the family Enterobacteriaceae. This family are all small gram-negative non-spore forming bacilli. They are also all facultative anaerobes, and they all can ferment the sugar glucose. The Enterobacteriaceae has 53 genera. 24 of these genera have been known to cause human infections. Oh, good. The lecture file is now finalized, so that should speed up my computer. Most of these genera, 12 of them shown here, are normal microbiota of the intestinal tract of many animals, including humans, and AKA we call these coliforms because we find them in the colon or in the intestines. They are usually opportunistic pathogens, meaning they do not usually cause disease, but they may cause disease, and they can cause urinary tract infections, pneumonia, septicemia, as well as other infections. The common genera responsible for causing opportunistic infections include Escherichia, like E. coli, or Enterobacter, Klebsiella, Citrobacter, and Serratia. Several of the genera of Enterobacteriaceae are associated with gastroenteritis, and these include organisms in the genera Salmonella, Shigella, and then certain variants of E. coli, such as E. coli serovar 0157H7. Normally, E. coli does not cause gastroenteritis, but E. coli serovar 0157H7 can. Any question about any of that? We'll also look at another family, Pseudomonaceae, which has members like Pseudomonas aeruginosa, which are similar to the family Enterobacteriaceae in that they are gram-negative bacilli, small gram-negative bacilli. However, the Pseudomonas do not ferment glucose, and they are all oxidase-positive bacteria. The family Enterobacteriaceae are all oxidase negative. The family Pseudomonas can be opportunistic pathogens, particularly Pseudomonas aeruginosa, which can cause infections in immunosuppressed individuals, burn patients, and trauma patients. Pseudomonas aeruginosa is also a healthcare acquired infection, and we talked about that earlier. We called it a nosocomial infection. Any question about that? So it's important for microbiologists to identify bacteria, gram-negative bacteria, and perform antibiotic susceptibility to figure out which antibiotic would be the best to give, and then identify the species. And to identify the species, you run biochemical tests on the gram-negative bacteria. So here are some of the common biochemical tests used to identify gram-negative bacilli. And we're not going to talk about all of these different tests, but we will talk about some of the tests, like the indole test. Some organisms can produce the enzyme tryptophanase, which converts the amino acid tryptophan into the products indole and pyruvic acid. So after overnight incubation, you add Covex reagent, which reacts to indole, and that produces a red band at the surface, which this picture is not showing very red, but normally that's redder than that. I think the picture is just a little off and it's showing it more purple. E. coli tests positive for indole production, so we'd say it is indole positive. Klebsiella oxytocca does does also, and Klebsiella pneumoniae does not produce indole. So it would be indole negative, which would be a yellow band. This is showing you the reaction, the amino acid tryptophan, when the enzyme tryptophanase is around, produces indole and pyruvic acid. The indole causes the red band. All right, let's move on to the methyl red test. The methyl red test and the VP test, or the Voges-Perz-Cure test, is usually started together, where one person inoculates one tube, and then the tube is split into two, with one portion used for the MR test and the other portion used for the VP test. So both the methyl red test and the VP test are used to detect the end products that result from glycolysis, acid end products or neutral end products. The methyl red test detects acid end products, and the VP test detects the neutral products from glycolysis. So glucose can be metabolized by glycolysis, producing acidic end products, and the methyl red test is used to find those. And then neutral products, where the VP test is used to determine the neutral products. So normally you inoculate an MRVP broth, and then you incubate it 2 to 5 days at 35 degrees Celsius. With the MR test, the tube is then split into two. One portion of the tube is used to detect the MR test. If the tube, you add MR methyl red to the tube, and if the tube stays red, that is positive for the MR test, meaning if the tube stays red, if acids are produced in the tube. And this is showing you an MR positive case and an MR negative case. The other half of the tube is used to test the VP test. And in the VP test, Barrett's A is added as a solution, and then Barrett's B is added, and then you shake it to get oxygen in, and then you let it sit for a while. And if red color develops in the upper portion of the tube within 60 minutes, then acetone is present. That's the neutral product. And we're looking for the production of acetone in the VP test. So this would be VP negative, and this would be VP positive. After you add Barrett's A and Barrett's B and you add oxygen, let it sit for 60 minutes. If it turns red, then this is VP positive. Any question about any of that? All right, so that's the VP test. We then do citrate test, and citrate utilization test is looking to see if the bacteria can grow on and metabolize citrate. Citrate is the only carbon source and the only energy source in this citrate tube. If the bacteria can metabolize citrate, remember citrate is in the citric acid cycle, then the bacteria will grow and the tube will appear purple if citrate can be utilized. If citrate cannot be utilized, the bacteria will not grow because it has no carbon source or energy source, and the tube will remain green. So that would be citrate negative and citrate positive. I believe E. coli and Shigella are, nope, I don't see which one is going to be citrate positive. Well, I remember, I said Micrococcus varians is citrate positive, and Micrococcus luteus is citrate negative. I'm sure I've got an example up here. It just says E. coli and Shigella do not metabolize citrate. All right, let's talk about the PR carbohydrate metabolism test. Remember, in the IMVIC, it stands for indole test, the methyl red test, the VEP test, and the citrate test. Let's move on to the PR carbohydrate metabolism test. We're looking at carbohydrate fermentation, and there will be one sugar in the tube, which will be a different carbohydrate. It can be glucose, lactose, sucrose, mannitol, or indole for our unknown project, but it can be any other sugar. And we're looking at the fermentation of that sugar to produce acids or acid and gas. If the bacteria cannot ferment the sugar, then the tube will stay reddish or pinkish, and then the fermentation of that sugar will be negative. So if we had glucose in there, this tube would be PR glucose negative. Remember, we call it carbohydrate fermentation when we're talking about all of them, but when we're talking about one sugar, we will call it by the sugar's name, like PR glucose. Any question about any of that? All right, if that sugar can be fermented, it'll turn the tube yellow, and this we will call A for acid. If the fermentation can occur with the production of gas, we'll label that AG for acid and gas, and this will be a tube having bacteria that produce both acid, yellow, and then the bubble in the inverted Durham tube, which is gas production. Phenol red is the indicator that is red or yellow depending on the pH. And if it's below, if it's slightly acidic, it will be yellow. And if it's not acidic, it will be red or pinkish. Any question about any of that? All right. Motility test is a very thin agar solution that is thin enough that the bacteria can actually swim in the agar. And so what you do is you stab inoculate the motility medium. And if the bacteria do not have flagella, the bacteria cannot swim. And the only growth you'll see is in the stab. If, on the other hand, you stab the tube, which we can actually see a little bit of the stab right there, but it was stabbed down to here, and the bacteria are very motile, then the bacteria will swim everywhere in the tube, and the tube will be cloudy like this. This is a clear tube, and this has bacteria growing both in the stab, and it's swimming out into the media, and it will be growing everywhere, so the tube will be cloudy. That's what happens when the bacteria are very mobile. If the bacteria are just slightly mobile, why did that come up? I did the paint. Why is that continuing to come up when I'm clicking here? Let me do a search because I'm not finding, there it is. and it's bringing up the wrong window. If the bacteria are slightly motile in the tube, we will get growth in the stab, but we will get flaring of the growth out from the stab. Any question about any of that? Oops, that went a little too far there. Let me erase that. And usually you'll have flaring on both sides. It may not be the same flaring. But it can be the same flaring. So you'll see something like that where the growth is more than just in the stab, it's also flaring out from the stab. And that you see if the bacteria are slightly mobile. If the bacteria are very mobile, like I said, you'll see the bacteria move everywhere in the tube, and you won't see a pattern like this. You'll see cloudiness of the tube. Any question about that? All right, Shigella and Klebsiella are non-motile. We only see growth in the stab. Serratia marcescens is motile, and it's very motile. You'll see growth everywhere in the tube. I don't remember one which has slight motility. I'm sure I could look it up, but I just don't remember off the top of my hand a bacteria that has slight motility. And you will not see it like this. You'll see flaring of the bacteria. All right, the hydrogen sulfide test is looking at whether hydrogen sulfide is being produced by the bacteria. And you can measure hydrogen sulfide production in a number of different tests. You can measure it in the triple sugar iron. How did that get reversed? Triple sugar iron. Let me save that. Or TSI tube. You can also see it in the SEMS tube, and the SEMS stands for sulfide indole motility test. And you can see it in the hectone enteric agar plates as well. In all three of these tests, you can determine if hydrogen sulfide is produced by blackening of the media or of the bacteria on the HE agar plates. Oh, what happened here? Lost my position. So I think that's a triple sugar iron tube, and it's been inoculated. No, that's not triple sugar iron tube. Maybe that's a SEM tube, and it's inoculated, and you see blackening. So hydrogen sulfide is being produced. Any question about that? If not, let's talk about the triple sugar iron test. The triple sugar iron test is running a couple of tests at the same time. You're looking at fermentation of glucose. You're looking at fermentation of either or both sucrose and or lactose. You're looking at growth of the bacteria anaerobically, and then growth of the bacteria aerobically, as well as the pH of the media, meaning you can get different colors. And then you're looking also to see whether hydrogen sulfide is being produced. With the triple sugar iron tube, what you do is you get your inoculating needle. Let me give you an example. And then you take the needle and you pick up some bacteria from a colony. You can roll it in the colony, or you can streak it to pick up bacteria on the tip, and then you inoculate that needle into the tube, making sure you go down about 2/3 of the way into the tube. This is to get the bacteria to grow in the inoculation stab, and this growth will be anaerobic because it's down in the agar. And then you take the needle or the stab, and then you pull it out and you inoculate it in the bacteria again, and then you streak the bacteria on the slant of the TSI tube. This is to look at growth aerobically, because this bacteria will be exposed to the air. You then incubate the tube and look at it, looking to see fermentation of glucose and then fermentation of lactose or sucrose or both. And looking for the production of hydrogen sulfide. You can also look for the production of gas. All right, the test is rather complex. With tube 1, there's no change in the slant. It's not yellow, and it's not black, obviously. And there's no change in the butt. That's kind of an orangish, reddish color. That means there's no sugar that's fermented, no glucose, no lactose, no sucrose. There's no gas being produced, and there's no hydrogen sulfide being produced because it's not black. We know there's no gas being produced because there's no cracking of the media. Let me blow that up. No cracking of the media, no lifting of the media, so there's no gas being produced. In tube 2, it's black on the bottom, so we know that hydrogen sulfide is being produced. There's no cracking of the media, no lifting of the media, so there's no gas being made. However, in this case, we know that one of the sugars, glucose, was fermented because the black only happens when this is fermented, and we can't see that the butt here is yellow, but that's because the black is hiding it. If we had looked earlier, we would have seen, before the tube totally became black, we would have seen in the lower portion of the tube yellow from the fermentation of glucose. You'll note that the slant is red, and the glucose was initially fermented, and if we looked earlier, the slant would have looked yellow. However, glucose is at low concentration. I didn't mention that before. There's only 0.1% glucose. The lactose and sucrose are 10 times higher. So if only glucose is fermented during the time of this test, the glucose would be fermented and then used up. When the glucose can no longer be fermented, the bacteria, rather than die, will metabolize the protein in the media, and that will produce a base, and that will revert the color here from yellow, bring it back to red, and as more and more base is released by the cells, it will eventually become pink, which it kind of is now. That's really pink here, but it's kind of pink there. So glucose is being fermented, that's why it's red here, and the base is black, but if we were to remove the black, the base would be yellow because the glucose is being fermented, and we can see the yellow in the base because anaerobically, the fermenter sugar is slower than aerobically, so that the base would appear yellow if we were to remove the black. And the slant is red because all of the glucose was used up on the slant, and that changes the color to reddish or pink. In tube 3, we see what's happening in tube 2. It's just that hydrogen sulfide is not being produced, so it's not black. And we see that there is yellow in the butt and the slant is red. So only glucose is being fermented. And that's why it's yellow in the butt, because glucose being fermented produces an acid, making it yellow. And the glucose is fermented slower in the butt than in the slant. So the glucose is still being fermented, it's still yellow. The slant initially was yellow. You have to look at it earlier. But with time, all the glucose gets fermented. There's no more glucose around. Rather than die, the bacteria start utilizing protein in the media. And the protein will release ammonia from the amino acids. Ammonia will be released, and the cells will release it, and that will change the color from yellow to reddish or pinkish, because ammonia is a base, a pretty strong base too. In the case of 4, we record this as A for the slant and then A for the butt, because it's yellow in both the slant and in the butt. And it's yellow in the slant because, yes, glucose is being fermented, because if any other sugar is fermented, glucose is always fermented. And then at least lactose or sucrose or both of the sugars are being fermented. And that's why the slant is yellow, because the lactose and sucrose are at 10 times the concentration of the glucose. So not all of the lactose and sucrose have been used up, and that's why the slant is yellow. This tube also shows cracking of the media right here, and then lifting of the media. And if ever you see either one, then you know that gas is being produced. So this would be A for the slant over A for the butt. And this one would be negative. They're using a K for the butt because K is alkaline. Excuse me, for the slant. Over A for the butt because the butt is yellow. Any question about any of that? And for this one, it's negative or K over K. Any questions about the TSI test? All right, let's move on to the sulfide indole motility test. Once again, this is running 3 tests at one time. It's running the sulfur production test, the indole test, and the methyl motility test. Motility test. How you inoculate the SIM tube is you get your bacteria on a needle, the tip of the needle, and then stab inoculate the tube, going about 2/3 of the way down the tube. See if I can blow this up. We can actually see the stab there, and it's a little less clear there and there, as the bacteria growing in the stab. All right, interpretation is multifaceted because you have 3 tests running on. In the control tube, we have no hydrogen sulfide being produced because there's no blackening of the media. There's no motility. I don't even see the stab mark, but let's assume you have a stab mark, or maybe they didn't do this because it's a negative, but there's no motility. There isn't growth outside of the stab. And then for the indole test, you add Kovacs reagent, and this is yellow, so that's Kovacs negative. For this tube here, we'll start with this band here. When we add Kovacs, the media turns reddish, and so that is indole positive. The tube is black, so that means that hydrogen sulfide was produced. And then you can't see it, but the bacteria are everywhere in this tube. And that means that this bacteria is motile because the bacteria grew outside of the streak from the stab mark. Any question about tube 1? In tube 2, once again, when we add Kovacs reagent, we get reddish reaction. So it is indole positive. There's no blackening of the tube, so it's hydrogen sulfide negative. And then the growth is only in the stab. So this bacteria is not motile. Any question about any of that? Tube 3, I think it's the same. Indole positive, no hydrogen sulfide production, and it's not motile either because the growth is only in the stab. Let me make sure that that's true because that's a little flared right there, but tube 3, motile negative. I got it right. Tube 4 is indole positive because it's red. It is hydrogen sulfide production negative because it's not black. And then let me see if I can blow this up. I can see it, but I don't know if you guys can see it. You'll note there's the stab, and the bacteria are growing outside of the stab because it's flaring. Tube 5 is indole negative because it's not reddish, it's yellow or clear. It's not motile because the growth of the bacteria is only in the stab. It's not flaring out or all the way over. And then it's hydrogen sulfide negative because there's no blackening in the tube. Any question about any of that? All right, let's talk about HE test first. With the HE test, you can test for hydrogen sulfide production. If the bacteria produces hydrogen sulfide, the bacteria will look black as shown here. If the bacteria do not produce hydrogen sulfide, they will not be black, as here. So this bacteria normally was whitish in color, and it's not black, so it's hydrogen sulfide negative. MAC and HE can also look at whether gram negatives can grow. If there's growth on a MAC or an HE, then the bacteria is likely a gram negative because the MAC and the HE have chemicals in it that inhibit the growth of gram positives. For the MAC, I happen to know the chemicals are, sorry, I'm drawing a blank on the name of that. Geez. The first stain of the gram stain is crystal violet. It has crystal violet in it, and that gets in the cell walls of gram negative cells. I mean, gram positive cells, and prevents their growth. It also has bile acids in it. Bile is in the intestine, and bile will not allow most gram positives to grow, but most gram negatives already grow in the intestine, so they're exposed to bile, and so they can tolerate the bile. So if you get growth on both the McCankey's and the HE plates, then that growth is likely to be gram negative because the gram positives are selected against. And it's not 100% accurate, but it's probably over 90% accurate, probably about 95% accurate. All right, both the McConkey's or the MAC plate and the HE plate can also look at the fermentation of lactose. If the sugar is fermented in MacConkey's, then the cells and the media around it will be pink because of the fermentation of lactose creating acids, and that acid environment will change this pH indicator to be pink or purple pink. If the bacteria can grow, it's a gram-negative, but if the bacteria does not ferment the sugar lactose, the bacteria will be its normal color. In this case, it's white or off-white. Any question about MAC? All right. For HE, if the bacteria can ferment the sugar lactose, the bacteria will change the environment, making it more acidic, and that will create a salmon color bacteria or salmon colored colonies. Salmon is the color of the fish salmon. It's sort of a yellow orange. Already mentioned that HE can determine whether there's hydrogen sulfide being produced. Oh, if there is not a salmon color, then the bacteria will be their normal color. And these are bacteria that do not ferment the sugar lactose. Any question about any of that? All right, let's talk about Laboratory 9 exercises. You're not going to be performing any of these biochemical tests because everything's going to be done online, but you are responsible for knowing how to perform and interpret them. View the following video clips on the carbohydrate fermentation test, on the SIM test, and the MRVP test, and the triple sugar iron test. We also have a video discussing the MacConkey's agar, so watch those videos. You'll also need to view some of the information from the website Microbuzz. Let's see if I can get that to open. And you need that information to fill in the table for the worksheet. You only need to click on the differential tests, and you only need to look at the tests that are in the table, because you need to look at the tests to get the information from the table. Most of these tests, if not all of them, are either tests you were exposed to today or you were exposed to sometime in a lab in microbiology. Did I hit the wrong button? I don't know how to back up with this browser. Let me try that again. Nope, that's wrong. Come on. So the MRVP test was discussed in this lab. Citrate test was discussed in this lab. Sims medium was discussed in this lab. Triple sugar iron was discussed in this lab. Blood agar was discussed earlier in the lab. So was the coagulase test. Motility test is discussed in this lab. Catalase test was discussed earlier. And the oxidase test was discussed earlier. I don't remember if we discussed the Bialasculin test earlier or not. I think we did. All right, any questions about any of this? All right, so complete the worksheet and the case studies. Let me show you the worksheet. For worksheet to lab 1, you need to fill in this table. Oxidase is in a previous lab. Indole, I think, is in this lab. MR, VP, and citrate, all of these are in this lab. Our carbohydrate fermentation, like glucose fermentation, we discussed in this lab. Gas produced of carbohydrate or glucose fermentation, we have discussed. For TSI, we discussed this in this lab. And Sim Auger Deep, we discussed in this lab. You'll need to talk about the lactose fermentation test, and that will be ran with the selective and differential media like McConkey's or HE. Any question about the table? Like I said, you will need to look up some information because not all of it is in this lab, but we have discussed it. All right, then in the worksheet, complete 2, read the question, and then fill in the answer. What reagent must be added to detect the presence of cells? So write your answer. And 2, and that is discussed in this lab, by the way. And then 2B, methyl red is a pH indicator that remains what color under acidic conditions. And then 3, actually it goes 2A, 2B, 2C. So answer these questions. Remember in 2D and 2A, you have 2 questions. You have 2D1, which is Simmons citrate uses the pH indicator, name the pH indicator, and turns the color, what color, under bold conditions. And this one is so if a neutral product is forming glucose, no, sorry, it's 2D. Simmons citrate uses the pH indicator, name that, and then tell me the color. If it's a blue color, when does that happen? Under acidic or basic conditions? And here you need to circle, underline, or make bold one of these conditions, and then submit your paper that way. 2E, what is the pH indicator used to detect PR, carbohydrate fermentation? So name the pH indicator. And this indicator is yellow under acidic or basic conditions. Select one of the 2. All right, so that's how you fill in these questions. I'm going to skip those. You can read through it while you're answering it. And then complete the case studies. The case studies are in the lab. I thought I made this one doable. Maybe it's in the module. Nope, must be in the lab. Or else I made a mistake. Oh, that's the wrong one.
I can see the case studies in the module.
Are they in the module online or?
Yeah, like the end of it. They're actually before the chart.
Oh, so I need to go up in the.
I think so,
Thank you.
at least from what I have printed out.
Oh, that's
So
the
like at the
worksheet.
end of the module is where I see the case studies.
That's where I was looking
There.
for. Oh, I didn't go to the, sorry, I
Yeah.
didn't go to the module. I thought I did. So the case studies, go ahead and read through it. I'll just go through case study 1. A prison inmate is brought into the ER room with an open wound, and this prisoner is known for self-inflicting. A routine culture and sensitivity test is ordered. So they grow his bacteria that's in the infection on a blood agar plate, and you can see it growing here, and you can determine the hemolytic pattern from this plate. And then they grow it on a McConkey's agar plate here. And you can see the color of the bacteria and what that means. And then they do a gram stain, which is shown here. You got to blow that up to see it. Blow it up really big, probably even bigger than that. That might work. Maybe that's too big because it looks a little blurry. Let me shrink it down to 300. All right, it's difficult to determine the shape here, but I think you can determine the shape. I'm gonna blow it up once again really big. Let's go even bigger. It's a little blurry, but you can determine the shape like these cells here and those cells there. Maybe that cell there too. And you can determine the gram stain really easy on that. So record that information, and then they did the catalase test, and it was catalase positive and oxidase negative. And on day 2, they did some other tests and got the test results. For A, that's the Sims tube with Kovacs reagent added on top. And then B is the MR test. And C is the VP test. And D is the citrate test. And E is the triple sugar iron test. You can notice an air bubble lifting of the media and cracking there. And then the butt is yellow and the slant is yellow. Remember, when you discuss the TSI, you should give the slant color over the butt color. So this would be yellow slant, yellow butt, or yellow/yellow. And you can record that as positive, or A over A, or positive over positive. Any question about that? And then with case study 2, put your results in the worksheet. Let's see if I can find it here. So put your results here for case study 1, what the oxidase test was, the fermentation of the glucose, fermentation of the lactose, fermentation of TSI, hydrogen sulfide production, if that occurred, motility test, the indole test, the MR test, the VP test, and the citrate test. Put your information here and then go up to this key. Only use this key for this lab. Do not use it for your unknown project. And then determine which species is causing the infection of case study 1. Any question about any of that? And then move on to case study 2. Let me shrink that a bit. Case study 2, you can read it. Mikey talked his mother into buying a turtle, and he's well at changing the water, but he's not so good at washing his hands after touching the turtle. About 2 weeks later, Mikey develops diarrhea, nausea, and fever. In his petition, orders fecal specimen for routine culture. And there you can see the bacteria growing on McConkey's agar and bacteria growing on HE agar. The arrows are looking at the bacteria of interest. And then here are the test results. Once again, write the test results in the table in the worksheet. And then use what is the test results with the key for identifying the bacteria. Any question about any of that? All right, it's a little before 7:30. I'll be here until 8 o'clock to answer any questions. But once the last student logs out, I am going to log out as well. So if you have a question, make sure you ask it while I'm still here. So try and do it early in the lab. Any questions?
No, and it's just me. So
All right, when
Um,
you, I'll
I'm the only one.